Okay, everyone, so maybe we can start. So good morning and welcome to this final MMIV seminar of the season before we all take our summer holidays. Today's set of talks is more consistent than usual, as we have three speakers are from the same group, which is very nice to get some coherent topic. The title being the imaging and the importance of cortical myelin in mental health. The first one of the speakers is Anders Lellevik Thorsen, who is a psychologist and a postdoc at the Center for Brain Plasticity at Haukeland University Hospital, also a researcher at the Crisis Psychology Center, UIB, and a guest researcher at Vanderbilt University Medical Center. Obviously has his hands full, so good that you have time to come here and speak to us. The second speaker will be Hilde Brekke, who is a PhD student halfway through her work, who some of us already had the pleasure to meet when she visited us at the machine learning group when she spoke about cortical myelination in OCD. And last but not least, the woman who maybe needs no introduction, but will get one nonetheless, Olga Therese Austdal, who is an associate professor at the Department of Biomedicine, University of Bergen, and also a consultant neuroradiologist, Haukeland University Hospital, and is an expert on she studies the physical underpinnings of mental disorders and their treatment. So looking forward to be lectured on that. So yeah, I think we can start.
Great. Yeah, thank you so much for the introduction and so much for the opportunity to present to you today. Especially since it's, as I gather, right before the holidays. So perhaps we need to keep it quite short. We are, as you said, all from the imaging working package of the Bergen Center for Brain Plasticity, which is also founded by the Tormoen Research Foundation, the university, the hospital, and as well the Kavli Foundation as well. We have, we have already been introduced sort of our speakers for today. And we are also so glad to have a whole bunch of students in our group who will also be involved in some of the projects that Olga Therese will be presenting in the last bit of this lecture. And we are going to talk about the imaging and the importance of cortical myelin, which is something that we have been working on now for about 1.5 years. And I would say that sort of, at least my understanding of it has grown and probably changed quite a lot during that 1.5 years. So I'm very happy to present it to you all. So first, what is myelin? So you can think of myelin in the central nervous system as this lipid-rich insulation around the axon of the nerve cell, right? That's often the thing that pops up in the textbooks. Now, this myelin has it should insulate this axonal wire, if you want to put it like that, and it wants to speed it up, to be as efficient as possible, and to have as little loss as possible. And this is supported in the central nervous system by oligodendrocytes. But perhaps that's not all that the axon or that the myelin does. So recent studies suggest that it's actually involved in many functions. For instance, providing energy for the neurons, regulating axonal growth, so that's sort of hindering, come in if you want to, hindering axonal growth, so that it's not growing too much, and perhaps also actually synchronizing action potentials around nearby neurons. So it might be that myelin does quite a lot of sort of active stuff, and doesn't only act as a sort of passive insulator. And when we think of myelin, we most often think of these deep white matter tracts in the brain, sort of the big wires. But that's not the only place where myelin is. So here's actually it's a histology using an electron microscope of myelin. So here you can see there's no pointer, is there? No, okay. So here you can see the axon, and then this the myelin sheet around it. So that's an example of something it's going correctly. It's insulating as it should. But this picture actually also shows that it's a hypermyelination, because you can see that there are not multiple layers around already myelinated axons. This is from a mice model. So an example of that, it can be too much of a good thing. And I was actually quite fascinated when I read about the history of cortical myelination. So already the Vogt and Vogt couple back in the early 1900s, they found that myelin was perhaps an important way to describe the organization of the cortex itself. And they described 4 different sort of types of myelination, which are differently related to the cortical layers. That's what we called myeloarchitecture. Interesting to know, their close collaborator, who's also shared a lab, Bodman, he instead focused on the cytoarchitecture, so the way of describing the cells of the cortex, which perhaps sort of won out and something that most of us have read a lot about, more about than myeloarchitecture. But in recent years, cortical myelin has received a whole lot of interest. Perhaps now, since, as we're going to tell you in a short while, it's available for imaging, and we now have reliable enough ways to image it. So we're going to go through a couple of ways you can do that. The first study, actually, who tried to image cortical myelin in live humans was already actually in 1992. So very close to when we started using bold fMRI. One of the ways that you can do it, it's also the way that we have done it in Vilde's study, is to compare the signal intensities in white and gray matter. So let's see. This might be difficult to see here. So then what we have done is then take the intensity value across several distances in gray matter, as well as in the white matter, and then you contrast these two. Where we know that sort of white matter has more myelin than gray matter sort of on average. And then you see sort of how similar or dissimilar are these tissues, where a high contrast means they're quite dissimilar, meaning that the myelin perhaps is not that high relevant in reference to other areas, where if they are very similar, then we think that myelin is higher. Another way to do it, which was described by the Human Connectome project, is to take the ratio between the intensity in T1 and T2 weighted MRI. This is a newer technique, and perhaps the technique that has resulted in this sort of big explosion of myelin imaging that we see now. I also find it interesting that when you read this paper by Glasser and Van Essen in the Human Connectome project, they're really sort of adamantly saying that this is myelin, this is a myelination map, which is perhaps not the case, as we will see a bit later. The good thing is that sort of regardless of which method you use, the maps seems to be very similar. So here on the left, you see a smoothed image from the PNC study. So this is mostly adolescents. We see that more myelin in this case is in regions which are in more red, compared to the Content 69 atlas for the Human Connectome project on the right. And as you can see, these images are very similar. And what I found sort of increases my trust in this method is that we get these kind of maps from many different techniques across many different studies. So we seem to be tapping onto the same sort of substrate. But we also have other methods, which I know that some of you are more familiar with than I. So we have magnetization transfer ratio, longitudinal relaxation rate, and also myelin water fraction, which are more sort of specialized MRI sequences that you can use. The nice thing about the previous sequences, of course, is that most of us already have data on there. We at least have a T1 image, and perhaps we also have a T2. And if those are of sufficient quality, then it's quite easy to get some estimates. What's good about these methods, and also the T1, T2 weighted contrast, is that you could also look at myelin in subcortical structures, such as the amygdala, the hippocampus, and so on, which, especially since we work in mental health, that's regions that pop up a lot and that we really want to image. And here it's important to know that all these methods that I just described have been validated through histology and sort of compared to each other. And what you find is that they all tap into sort of myelin to some degree, but not to a perfect degree. So they are sort of imperfect approximations, right, of myelin. So the things that I've talked about so far, we're going to talk about some more. It's not myelin per se, but it is close. But there are sort of also other confounders that might influence these signal intensities that we are using. So that brings us to sort of what's the use? It has now been shown for quite some years that myelin in the cortex seems to be related to symptoms and disorders of mental illnesses, and actually many different kinds of problems. And has been mostly studied, I would say, in patient populations in schizophrenia and bipolar disorder, but also to some degree in PTSD. And now to an increasing degree also in normal populations, so seeing how it maps onto symptoms in a sort of population-based samples. But it's also interesting to note that there are a lot of disorders which have not been investigated. So OCD, which we're going to talk about shortly, this technique has not been used there. There we have mostly focused on diffusion-weighted imaging and these deep white matter tracts. And the findings are not always consistent. So I think there's still a lot to go in this field, both to improve the methods, how we image myelin, and also making actually use of it in psychiatry, in mental health. And I think there we move on to some data, the more nitty-gritty. Villa?
Yes, so then I'm going to put what Anders has now introduced you to more into the context of OCD. So we are going to start with setting the stage of what we're going to look at this next part. We're going to look at why cortical myelination in OCD. Also, we are going to look at the methods that we applied that Anders has briefly introduced you to, and our preliminary results and conclusions. So first off, why cortical myelination in OCD? Why do we even think that this has anything of importance or is relevant to look at? And that's because we have looked at the literature. Like Anders said, no previous studies have looked at cortical myelination in OCD, but we have indications from the literature that this might be relevant. So like Anders said, there are inconsistencies in the findings, but generally we find that there are alterations in white matter, and that we see both in decreased volumes of gray and white volumes of matter. And we see in genetics that genes related to the expression of oligodendrocytes are altered, and that this correlates with the volume in the respective sites. And finally, more and more multi-site studies are also trying to tackle these difficulties with the inconsistencies, both in the small sample sizes, which are typical for clinical studies, but also in terms of applying harmonized pipelines. However, most studies indicate this decreased white matter. And when it comes to white matter tracts in OCD, these are widespread decreased. So on to our methods. We are trying to tackle this issue with small sample sizes by using data from a multi-site consortium, the Obsessive Compulsive Brain Imaging Consortium, is the precursor of Enigma OCD, which some of you are familiar with, the OCD consortium, that is. So in our sample, we have data from 8 different sites and countries, and we have applied harmonized data processing. And that means that we are doing a mega-analysis. We are performing all our analysis locally, performing the same pipeline. It obviously has some limitations and challenges to use different sites, and I will come back to that. So for our methods, like Anders was saying, we extracted gray and white matter signal intensities in one of the very first steps of the free surfer reconstruction of cortex before it's normalized. And then we performed the rest of the cortical reconstruction. We extracted these values, as Anders explained, in this contrast formula, in which the intensity contrast in the different region voxel-wise is fed into an independent component analysis. Before we did this, we also did an extensive quality control, which we then looked specifically at topological values in the measure of Euler number and surface holes, which is generated by ASEG in the cortical reconstruction by a free surfer. And then finally, we fed this contrast into the independent component analysis, and then we assessed the CASA results from this. So like Anders said, high contrast could indicate more similarities, which again then would mean more cortical myelination and vice versa. So these are the results from our ICASO of the independent component analysis. And we found that with 7 components, that was the optimal model fit, as you could see by the stability and the clustering and the ranking as well. So what we found was that there was a group difference in the loading to what was our 3rd component. We also did a leave one site analysis, which replicated the findings, except for when we left out the Barcelona sample. And we want to give a thanks to the Lundervolls for suggesting to include this analysis. When we look closer to the numbers of this non-significant finding in the Barcelona sample, we wonder if this might be due to a power factor, as this was the largest single site that we had. And yeah, I'm going to leave it to that. So this is our 3rd component. And what you see here is that this reflects that the pattern is that there is a lower contrast in occipital regions, while it's a higher contrast in superior frontal regions. And we found a group difference there with patients loading lower to this pattern than controls. These are both typically highly myelinated regions. And when we had a closer look, we actually found that the contrast in patients were lower in occipital regions. And this indicates more myelination in patients in these regions compared to controls. We also run a clinical characteristics post-hoc study to see if the component loading was associated with age of onset, as age of onset have indications of a genetic component. We also associated it with medication effects, which is a recurrent finding in OCD. And we looked at comorbidity, both depressive and anxiety disorders. We did not find any associations with that. However, we found associations with sexual and aggressive symptoms. We also found that age and the quality metric surface holes was important for the model, the statistical model. However, that the group effects remain despite controlling for this. So what I forgot to say was that OCD is characterized by 2 sets of symptoms. The symptoms of obsessions and compulsions. They usually come in sets, but they vary greatly in their expression. And the obsessions are characterized by impulses or urges, mental imagery, while compulsions are acts that are performed in the aim to neutralize the discomfort by the obsessions. So they come in sets like sexual and aggressive and contamination and washing, for instance. So the summary that we have, our preliminary summary, would be that there are group differences in the loadings of these cortical myelination patterns in our sample, and that we see indications of patients showing more cortical myelination in occipital regions, and that this supports an idea of investigating further the role of cortical myelination, not only in OCD, but further in psychiatric disorders. And from here, it's Olga Therese next.
Okay, so we are actually ahead of time. Means more time for question, I guess, and discussion afterwards. Okay, just let me figure out the technical part here now. Great. Okay, so I'm going to then talk about future studies. So where do we go from here based on what we have found so far? So what would be the natural next step for us? And that's something that we've thought a lot about, and I'll give you some clues today what we are thinking about to do. So let me start by this. So there is something that many researchers in psychiatry have thought about more recently, and that is that psychiatric disorders, they mainly target youth. So this figure here, it illustrates the age of onset of most common mental disorders. And in contrast to major neurological conditions, which are most prominent in the aging population, psychiatric disorders target children, adolescents, and young adults. Indeed, 50% of the patients, they develop symptoms before the age of 14, and 75% before the age of 24. So, and the same is true, although OCD is not depicted here, the same can be said about OCD, which 1/3 develop symptoms before approximately 15, and then the remaining before the age of 25, mostly. So together with the evidence from genetics and other epidemiologics and so on, there are now increasing evidence to support that these disorders, they do have a neurodevelopmental etiology. So if we then want to understand why and how someone develops a psychiatric disorder, we need to study brain development. And that also includes the development of cortical myelination. So cortical myelination is a continuous process during childhood and adolescence. It is very protected in humans compared to rodents and more primitive primates, and it continues and it extends into adulthood. Indeed, some studies show that it continues into the 4th decade of life. And it's illustrated here in this figure. It has a posterior-anterior gradient, indicating that the more posterior regions, they myelinate before the more anterior regions. And on top of this, there is a regional specificity, so that more sensory motor regions, they myelinate before the more complex cortical regions involved in emotion and cognition. So in the beginning, we think of this cortical myelination can be ascribed to the oligodendrocyte and wrapping around the axon, so the myelinization per se, and later in development, it's due to thickening of the already existing myelin sheaths that increase the myelin that we see, at least in neuroimaging. And this is reflected then in both charts of gray-white matter contrast and in T1, T2 charts or images. And it turns out that there is if you look at the GVC, there's a decrease with increasing age. And for the T1, T2 contrast, you will see an increase in the whole brain signal with age, indicating that there is an ongoing myelination. So it seems to be the case, as indicated in this study from 2019, that this pattern of cortical myelination in adolescents is different for youth with symptoms from anxiety or prodromal psychosis. Indeed, as indicated here by the figure, they have regional increases or decreases in the gray white matter contrast compared to healthy peers. So, of course, this is very speculative, but that may indicate that if your cortical myelination process go awry in childhood or adolescence, well, that might put you at risk of developing a mental disorder. So then if we want to understand more about how we then develop an aberrant cortical myelination, we need to look at what determines the myelination process during development. And as for the rest of the brain development, this is, of course, the development is determined by genes, but it's sculptured by environment. And of course, gene and environment will always interact, and it will interact perhaps differently at various ages. And we know that a significant amount of our genome is devoted to brain development, and it is expressed during childhood and adolescence. But as I said, although it's sort of determined by genes, it's sculptured by the environment. And the environment can act in two different ways. It can sort of decide the end point of the developmental process. So whether you will have decreased or more myelin when you become adult, or it can impact the trajectory. So sort of the pace of cortical myelination. Will your myelination process go faster or slower compared to age-matched peers? So when we talk about environmental influences during development, those that can influence brain development, there are at least one group that is highly relevant, and that's the one that I'm citing here, and that's early life adversities. And why is that so important? Well, we know that approximately 50% of mental health problems that emerge during childhood and 30% during adulthood can be related to the experiences of early life adversities. So this is a significant contributor to our development of mental illness. And there are several types, and of course I can't go into every one of these, but they are usually, when they describe it in the literature, they describe it in two categories, one being threats, so harm or threat of harm to the child. It can be abuse, violence, natural disasters, war, and the other one being deprivation, so the lack of the expected inputs from the environment. That could be a lack of social, intellectual stimulation. And why do we think this is important for cortical myelination? Well, it is the case that if the developmental process is ongoing, it will be more dynamic, it will be more sensitive to adverse events. The influence from environment. And indeed, during development, several brain regions, including a part of the frontal lobe, contains higher number of cortisol receptors. So we know it's more sensitive to these stressful events during these life phases. So what do we know from the literature then on early life adversity and cortical myelination? Well, most of what we know is based, of course, on animal models, which is, of course, you can more control what you expose the animal to and follow it with very regular intervals throughout the development. And it's shown that rats who are exposed to stressors early in life, well, they will have less cortical myelin in some regions, perhaps mostly in the prefrontal cortex when they become adult. And what is the reason? Why is that the case? Well, we don't know, but there was a recent, very interesting study published in 2020, which they showed that the oligodendrocyte that produce myelin, as Anders said, They are initially progenitor cells, and then they develop into the mature oligodendrocytes. And in pups that were rat pups who were exposed to stresses very early in life, there was a precocious development from these progenitor cells into adult cells. So in adolescence, these rats expressed more of the adult oligodendrocyte population. And what was a consequence of this was that when these the pups became adult, the progenitor pool of oligodendrocytes, it was depleted. So meaning that they had less oligodendrocytes to recruit if needed. So they could produce less myelin if needed by the brain, to say. So this is from the animal literature. What do we know then in humans? Well, that has actually not been studied. So we don't know how these early life adversity affects human cortical myelin. But what we know is indirectly is from studies where they have looked at children who grew up under low socioeconomic status. And unfortunately, at least in the US, if you experience to grow up in a low socioeconomic status environment, you are also more prone to be experiencing stressors. And in these individuals, they have shown, youth living in under low socioeconomic environments, they express less cortical myelin compared to peers that grew up in more high socioeconomic status homes. Unfortunately, of course, this is during development, so we don't know the end point. It might be that this is something that is transient and will be later corrected. So, so then what we want to do is we want them to study in a future study now how early life adversity may impact this trajectory of cortical myelination. And to do this, then we need a huge database. And we are so lucky to have access to the ABCD study, or data from the ABCD study. And the ABCD, which is short for Adolescent Brain Cognitive Development, it is the largest long-term study of brain development in the US. It is it's a NIH-funded study, including 12,000 children recruited when they were 9 or 10 years, and then they're going to be followed for at least 10 years. And they're going to do repetitive MRI scans, and we also collect a lot of other types of behavioral clinical data, biospecimens, and so on. And these researchers then they track both the behavioral and the brain development throughout adolescence and into adulthood of these children. And at present, they have now released the first and the second MRI acquisitions of data from the first 2 acquisitions. And we plan to avail ourselves and use these data sets to answer or to tackle this question. So the ABCD, they have very thorough characterization of the early life adversities that these children have experienced, so both threats and deprivation. And we can use then the MRI data. That's a multimodal MRI, and they have both a T1 and a T2 to either calculate the T1, T2 contrast or the GBC as presented by Anders. So we want to investigate this both cross-sectional and longitudinally. So we know, as I said, that early life adversity is associated with a number of mental illnesses, but the association is especially well established for internalizing disorders. So what is internalizing disorders? I like to call these disorders emotional disorders, but I know that might not be the correct term, but that's disorders involving emotional problems. So the first one is anxiety disorders, where you experience excessive fear. And in here, I include a post-traumatic stress disorder and OCD. And then you have depression, where you experience, sort of say, too much sadness. So both involve emotional problems. So these disorders, they have overlapping clinical characteristics. They both emerge during childhood and adolescence, and they have a common risk factor being early life adversities. And importantly, they are very often comorbid. So if you suffer from depression, you are more likely also to suffer from an anxiety disorder and the other way around. Then we know, as I said, that these internalizing disorders, they share clinical characteristics and they have a common risk factor. Then we can ask ourselves, well, what about cortical myelination in these disorders? Will that sort of reflect the common risk factor? So will it reflect the early life adversity and be common across the disorder, or will it be disorder specific? That is something we do not know and would like to look at in the using the ABCD sample that I just described. And sort of that might tell us more about the pathophysiology. So is this an early feature, perhaps reflecting the common risk factor, or is it something later on reflecting more the sort of the specific symptom structure of each disorder? Yeah. So just to add, it's important to know that the ABCD study, it also includes youth that fulfill the diagnostic criteria of an anxiety or a disorder or depression. So we can study this using this population. Okay, so we've actually come to the summary. And what Anders just said in his part is that we can use various MRI techniques that are sensitive, but not specific to cortical myelination. And that is important, the not specific part. It's a proxy, but it still allows us to do some interesting preliminary work, and then perhaps we will have to go on and verify it either in animal models or using different techniques to confirm. And as Vilde said in her part, cortical myelination may be altered in patients with OCD, reflected in the GVC contrast that we studied. And then I outlined some future work in which we especially focus on early life adversity and how that might impact cortical myelination. And that we know from animal models, but we don't know the consequences for mental health. So it may be the case that this aberrant cortical myelination is only in youth with symptoms of, for instance, internalizing disorders, or it might be a more general feature track due to the experience that these children and adolescents had. So with that being said, we would, of course, like to thank a lot of people. So here we just listed those that we have collaborated with in Vilde's study, and that is Dag Alves and Lars Vestli at University of Oslo, and Odile van den Heuvel, who is the co-PI of the neuroimaging work group at the center. She's based in Amsterdam. And then, of course, those who contribute with data, the OCD Brain Imaging Consortium. And as Anders said, we are funded, the Bergen Center for Brain Plasticity is funded by Kavli, Trondheim, University of Bergen, and Haukeland University Hospital. And we are very happy to be here today. And of course, we welcome collaborations from people here. And we have already established some and hope to establish more. So thank you for inviting us. Okay.
Yes, so if anyone has any questions, now is the time. We have quite a lot of time left, so please. Yes.
Yeah, first, thanks a lot for a very interesting overview of the field. I think it's very fantastic what you have established and the way you utilize data from external, all kinds of sources to really scrutinize and assess your hypotheses and study that. That's really impressive and very nice. I was wondering, do you have any expectations when it comes to the 7 Tesla system for depicting these very subtle, probably differences across patients or study persons? Do you have any comments? Whether you think that could make it easier to?
I will then only speculate, of course, and we might contribute to say something more add on to this, but of course we know from at least from fMRI, and I guess it's the same with T1, then you can get closer to the cortical layers with 7 Tesla. So of course that would be amazing because we know it's not evenly spread out, the myelin and cortex. It's more in deeper layers, as Amanda said. So of course, if we could go directly into the deeper layer, then we might get a more sensitive measure. I think that would be a big advantage if we could go for 7 Tesla.
Yeah, that's not what has been used in the
No, it's 1.5 and 3.
Yeah.
So it's possible to do with the existing, at least it looks like. Yeah.
Absolutely. Yeah, absolutely. So the people behind the Human Connectome project, of course, really argue that you should have as low resolution as you can get voxels, so that it's possible to do it as of the typical 1 millimeter voxels. You can go lower, so it's
So there's also a question here from Carsten Specht, a question for Anders. Perhaps he already mentioned it, but he needed to pick up a phone call. But the question is, did you also look into the voxel-based relaxometry, VBR, based on T2 relaxation times?
no. Should I answer it there, or is there a microphone, or is there a?
I think if you speak out loud, it should be good, because we already, yeah.
No, I did not. So I realized that there are a ton of different methods of how you can estimate it. Most of the time, I'm not very familiar with, to be honest. But yeah, we have focused more on the ways that are so readily available from sort of typical MRI sequences. Yeah. So now, but I think I don't know, much more clever people than me can probably do some interesting things with the scanners that we have available to us here as well. So.
Just another question.
Yeah, this is very interesting. I learned a lot. Thank you very much. I have a question, or a couple of questions. So, to what extent is this myelination dynamic during adulthood? And do you look at cross-sectional data, side differences? So, would you expect, or have you seen longitudinal studies do this change?
I could just answer for development. There's one longitudinal study, so there's very few. So this is general. I was quite surprised when we started to do this. Then there are so few studies because it's available in T1 image. You could do it. There's so few studies who have done this, and especially in longitudinal, because it's probably going to be very. If you look at development, it's going to be very different longitudinally compared to if you use cross-sectionally. And when it comes to adults, so did you think of healthy population of patients?
Actually, anything, because I also think about, we have lots of gray matter changes in CT, so we should probably do this.
Yeah.
And see if.
I already thought about that.
I'm sure you have already suggested this.
Yeah, I did for a while ago, but yeah, don't think we have enough long-term data. Yeah, but when it comes to adult, I know they've done it in some patient population, but how much? And of course, they've done it in aging, but that's work done like with Anders Fjell, and they use this technique, but that's a while ago. Yeah,
I'm also not aware of much longitudinal research, especially in terms of using interventions. So I think that's something that we will want to look into both in our own data and really hopeful that some other of you have data can use. And I think you also of course mentioned gray matter alterations. So one paper by Frances and colleagues in 2009 actually suggested that when you adjust for the cortical myelination or the gray white matter contrast, then you might actually be more sensitive to alterations in cortical thickness, for instance. So that's, I think there are some interesting things to be done with these myelination maps. We were not able to replicate that finding. That's sort of when you adjust for it, then you get more specific cortical thickness findings. That's interesting.
I also did not find any age by group interactions, but there were some tendencies for age groups in general. That was interesting, but we didn't have the power or not within the scope of this.
Yeah, I just follow up this 7 Tesla possibility, because you have this, as you mentioned, the layered neuroimaging possibility now, and there are data repositories with data and also very good modeling. So you get a very good insight how the cortex might, the architecture might be by maybe going in this direction, and possibly you could also use the animal scanner, that's 17, because then you can also directly measure by multi-annular omics, proteomics approaches, the myelination in place. So, but I think it's very, very interesting, and it has to do with the wiring of the brain. Actually, you approach it by a proxy, by this myelination encoded, but it has to be propagation of information maybe, and that could be really difficult at one step further and look at the bold or other mechanism for functional processing locally within the cortex.
So yeah, so we didn't mention that, but that's also a part of the stress study with cortical myelination. Of course, I discussed we have a myelin expert at Institute of Biomedicine, and so I discussed with him. So what is to determine the cortical myelination or the myelination in general? And as he said, well, the neural activity is going to be very important for myelination. So would it be possible? That's never been done, but it would be interesting to sort of, can we link functional MRI, which is a proxy, everything is proxy in MRI, proxy of neural activity to myelin maps. That way, sort of look at what is determining the myelination. But that will require even more work and effort, because we have to combine different MRI techniques and skills. But it would be interesting.
It already makes quite a lot of sense with the map you have there, because the occipital and motor cortex are highly connected, and they're also used a lot. So
I have a question on the side with the early life adversities. How is that recorded? Are there behavioral clues they record? Because these children, they're in a voluntary study, and you can't just go ask them, are your parents depriving you? Or
no, I know this is really difficult to measure. But so the way we do it today is that it's based on questionnaire. So either the child report itself, or there may be sort of clues from the family reporting that, you know, there's one of the parents suffering from or having a substance abuse, or so it's very difficult to measure indeed, and some people think that if you look at socioeconomic status, that might also reflect it, and so on. So I know it's difficult. It's much easier to sort of experiment and manipulate it. Or you can do as of course, if it's when it comes to natural disasters or war experiences, that's sort of, then at least you can confirm it to some degree. But based on things you experience at home, of course, that's only your self-report. Yeah.
Or if we can have a randomized trial of child deprivation, it would be good for science, but not good for people.
Maybe corona. Yeah. To some extent, yes.
Okay, so if there are no more questions, then once again, thank you very much for speaking to us. Yeah, so the night is still young, so there will be some waffles and refreshments up in MMIV, so please join us, those who can and want to.